Hello and welcome to our channel. Today we are going to introduce a new series on genetic engineering. And on today's video we are going to focus on introduction to recombinant DNA technology and DNA cloning. So first of all, what is recombinant DNA technology? Recombinant DNA technology, which is also popularly known as genetic engineering, is a set of techniques which enables DNA from different sources to be identified, isolated, and recombined in order to introduce a new character into an organism. For example, if we see a wild plant which can withstand insect attack, genetic engineering allows us to identify a gene responsible for such resistance and transfer it to other plants which do not show resistance. This technology first came into light with the discovery of restriction enzymes by Swiss microbiologist Werner Arber in 1968. We are going to talk more about restriction enzymes in our upcoming videos. Many scientists have contributed in this field, but one scientist who is famous for his pioneering work is Paul Berg. He was the first scientist to create a recombinant molecule containing DNA from SV40 and lambda phase. Later, Boyer and Stanley and Cohen developed recombinant DNA technology, showing that genetically engineered DNA molecules may be cloned into foreign cells. Now, let's talk about DNA cloning. It is a very important aspect of recombinant DNA technology or genetic engineering. But before we dig any further, let's take a look at what clones are. Clones are genetically identical clusters of individual entities or cells that are descended from same ancestor. A very popular example of clone is Dolly the sheep. Similarly, DNA cloning is a production of identical DNA molecules from a single ancestral DNA molecule. In theory, a desired DNA fragment is selectively amplified, resulting in a large number of copies of selected DNA. But in practice, it involves multiple steps and rounds of DNA replication. There are two different types of DNA cloning approaches which are being used: cell-based DNA cloning and cell-free DNA cloning. So first of all, cell-based DNA cloning. This is the first form of DNA cloning to be developed, and it is an in vivo cloning method. That is, it is done with or within a living organism. And it basically involves the following steps, that is construction of recombinant DNA molecules, its transformation, then selective propagation of the clone, and then isolation of the recombinant DNA clones. The first step is construction of recombinant DNA molecules. Recombinants are hybrid DNA molecules that consist of autonomously replicating DNA segment, cloning vector or replicon, which are capable of independent DNA replication, and inserted element or gene of interest or desired DNA fragment which we want to transfer. This first step of creating recombinant is an in vitro method, that is, it is done outside the living organism. This step includes cutting the desired DNA and replicon using the specific restriction endonucleases, and then joining the fragments with ligases. All the enzymes for DNA manipulation would be discussed in the upcoming videos. The next step is transformation. In this, the recombinants are transferred into the host cell, either bacteria such as E. coli or yeast. This process is called transformation. Then the replicons undergo DNA replication independently of the host cell chromosome. After transformation, some host cells take up the construct and some don't. Hosts that take up the plasmid are known as transformed cells, and host cells that do not take up the plasmid are referred to as non-transformed cells. The next step of selective propagation of cell clones is very important because in this step, transformed cells are selected from the non-transformed cells. Every plasmid contains a specific site known as antibiotic resistance gene, which gives bacteria the ability to survive even in the presence of specific antibiotics. Therefore, when plated on an antibiotic selection media, bacteria which do not have the plasmid construct will not survive, and the bacteria which have the plasmid construct will give rise to the colonies. After selection, two steps are performed. First, these transformed cells are plated on agar in order to encourage the growth of cell colonies, and these population of identical cells are known as cell clones. Second, these individual colonies are picked up from the plates and then they are suspended in the liquid culture. But this doesn't necessarily mean that these colonies will contain the right plasmid. That is because during ligation, different fragments don't always get pasted in the way we want. Sometimes they are inverted and sometimes no desired gene is interested in the vector. Therefore, methods like restriction digestion or colony PCR can be conducted for the selection of recombinant DNA. The last step of cell-based cloning is isolation of recombinant DNA clones. The recombinant DNA clones are harvested from the suspended cells culture and then they are selectively isolated. So now let's summarize the cell-based DNA cloning. The cloning vector and the target DNA is ligated to form constructs. They can either be recombinant if they have the target gene or non-recombinant if they don't have the target gene. Then they are introduced into the host cells, and here three different scenarios can occur. One is that no construct is transferred to the host cell. They will be referred to as non-transformed cells, or they either contain the recombinant or the non-recombinant cells. Now they will be selected for the transformed cell, and it is usually done via antibiotic selection. Once the transformed cells are selected from the non-transformed cells, it is very important for us to select the transformed cells with the recombinant DNA. This process can be conducted via restriction digestion or colony PCR, and once we have the cells which only contain our target gene, it can be further amplified. Now let's move on to our next approach for DNA cloning, that is cell-free DNA cloning. This approach of cloning is entirely in vitro, and polymerase chain reaction or PCR, which was developed in 1983 by Gary Mullins, is a common method used for the same. It allows selective amplification of the target DNA sequence using specific primers and enzymes. It is a rapid, inexpensive, and a simple method of copying DNA sequences, but it is not suitable for highly adaptive or large sequences. This is all for today's video, and in the upcoming video we'll be discussing about enzymes for DNA manipulation and mainly about DNA polymerases. And if you like our content, please like, subscribe and share. Do follow us on Instagram, and if you have any queries or suggestions, please write to us on the given email address or in the comment section below. Thank you.